Hey future doctors, welcome to Dentistionary. Here you will be provided with information for the doctors from the doctors. Today we will be discussing about the last part of the importance for the subject biochemistry. So let's start without wasting any time. First of all, the chapter plasma proteins is what it was left behind in the previous video. So we'll be discussing it. From this chapter only albumin comes. And for the albumin, you have to write its function. This is the main thing you have to write. With this, you have to write the serum level, normal serum level in your blood, the sources, okay, and what will happen if you have deficiency of it. These 4 points are very main. In the functions, you can write colloid osmotic pressure of plasma, how it maintains it, transport function, nutritional functions, blood-brain barrier functions, edema functions, how it regulates edema. You can write all these. Okay. Then next, biochemistry of teeth and caries. From this chapter, only fluoride comes. Okay. Fluoride has very much importance in your professional exams. For fluoride, the functions are very important. How it prevents developing caries and mainly how it causes fluorosis. Means you have to write the normal values of the fluoride in body or in water you drink, and what will happen if the quantity of the fluoride in water increases. That what causes fluorosis. Okay, then write about fluorosis, how it's dangerous for our teeth, and how it can be prevented or treated. Okay. Next chapter is nucleotides, the structure of purines and pyrimidines. It's very easy. You just have to draw the structure and just define it what is in it. Okay. Then de novo synthesis of purines and de novo synthesis of pyrimidines. You just only have to draw the cycles in that de novo synthesis. In cycles, you can include, for the de novo synthesis of purines, you can include the general introduction, how it can be synthesized, then the preparatory step, then the main de novo synthesis, formation of AMP, and the conversion of IMP to GMP. Okay. For the pyrimidines, you can include how it differs from the purine cycle, and then the regulation and the synthesis and degeneration and all. For gout, you have to write about its general introduction, then what is primary and secondary gout, or primary gout and secondary hyperuricemia, then the clinical findings and the treatment. That's all for the gout. Let's move ahead. DNA and its replication. You can recall it from your 12th class. The structure of DNA, Watson-Crick model, the double helix model for the DNA structure. You can draw that and define it, how it can be made and how it works, how the double sulfur bonds and all hold it together. You have to describe all of that. Then the replication of the DNA. It contains, it includes many steps. Uh, the DNA polymerase, how it acts. You can define different types of experiments like Michelson-Stahl experiment, replication fork. You can describe all of these. You can draw diagrams, okay, and you can define that. You have to write all about the enzymes that are taking part in this cycle. You have to write it from the initiation to the primer and the elongation of the DNA strand. Then the how the termination works for that. Then the lagging strand and the Okazaki fragments. Okazaki fragments are very important in this. Okay. Then comes the tailing and capping. That's all for the replication. Now the transcription, the different types of RNA and what is RNA, the RNA structure, how it differs from the DNA. Okay. then different types of RNAs, then the transcription process, in which it includes initiation, elongation, termination, tailing, and capping. You can also describe it from the book of NCERT for the 12th class. It's just similar, but these have more explainable terms than the 12th class. So you should write those terms for the good marks. Then the reverse transcription. It is very important for your further classes for next years. So you should remember the reverse transcription, how it happens. For the tRNA, you have to draw the clover leaf model for it and describe it how it was made. That will be very important if it comes, and it generally comes in the short notes. For the transcription process, it can come in long answer type questions. Now the translation, it's just the process is important, and it generally comes in long answers, but sometimes it comes for the short answers for 5 marks. Okay, and then you have in 5 marks you can just draw the cycle and define it just like that from few points for few points. But if it comes under the long, you have to describe all the steps with the cycle, with the diagrams. Okay, in 5 marks you can only just draw diagrams. Gene expression, you have to define the mutation and the types of it. It has different types, point mutation, deletion, and all, and how it affects the general body or the development of the body or a child. Then the regulation of the gene expression, lack of one, it's very important, and it always comes in a short note for 5 marks. Then comes the liver function test. It contains these are all comes under the heading of liver function test. So it can come in long answer type question or short answer. In short answer, you just enumerate it and describe just like this that you can see in the slide, how the bilirubin measurement for the excretory function, the normal serum bilirubin, how it affects the urinary bilirubin, the normal values and the elevated or decreased values, then test based on the synthetic function of the liver, serum albumin level, prothrombin time, alpha fetoprotein. Then the test based on serum enzymes or the liver enzyme panel, which indicates the hepatocellular damage, markers of obstructed liver diseases like alkaline phosphatase, gamma glutamyl transferase. You have to write all these in that one heading. Okay, and you come up to 8 marks or the 10 marks. The renal function test is generally comes in the short notes. In short notes, you can define it in general terms, and for general terms, you have to enumerate the steps, the different kinds of steps based on function, the GFR measuring and tubular function, and on the clinical basis, routine clinical tests are there. The complete urinalysis, non-protein nitrogen test, serum electrolyte test. Then comes the marker of GFR, the clearance tests, and then for the proteinuria, we have to test the markers of glomerular permeability. These all comes under the renal function test. You just have to define them in the short or the brief form for the short notes. Okay, that's all. And markers of tubular function are also there. Urinary and plasma osmolality, concentration and dilution test to assess renal acidification. That's all. Thank you so much for listening to us to this video. I hope you found this video informative. If you want the PPT, follow the link below. And if you have any doubts or queries about this video or any other video, you can comment down below. We will try to solve it as soon as possible. Thank you so much.